Okay, so with the chromatid being a single chromosome and the sister chromatids being stuck together, also making a chromosome, is that X-looking chromosome just two sisters? Well, yes. When you actually see the X as two sister chromosomes, because back in interphase, when you are duplicating the DNA, they're all outstretched. They're not condensed. They're in the uncondensed form. So they're chromatin. And so the chromatin is when it's uncondensed and when it's able to be replicated, because it's not wrapped around the histones and into the nucleosomes and things like that. And then once it is replicated, so that means that you have identical copies of each strand of DNA, and you start to enter prophase because you're leaving interphase, then that's when the chromosomes condense into tight packages so that they're easily pulled across the cell and not all tangled up like a ball of yarn. So when you do see the X shape, it is only in nuclear division, so either in meiosis or mitosis. And when you're entering mitosis or meiosis, you've replicated the DNA, and so you have the identical copies of DNA. And so when you see the X form of the chromosomes, it's when they're condensed in nuclear division and they are sister chromatids. And here again you can see a blown up picture of that. So for some of the questions that are asking about amounts of DNA and how many chromosomes there are, so this is one of the ones out of your exam. If a somatic cell from a mouse contains 40 picograms of DNA during G2, so that would be an interphase, and G2, remember, is after replication. So if it said G1, that would be a whole different story because it's before DNA replication. So you got to pay attention to whether it's before or after S phase. And G2 is after S phase. So the DNA is replicated. You've got the identical copies of each of the chromosomes. And so now you're going to go into meiosis, but you're just going into anaphase 1 of meiosis. And so leaving interphase, entering prophase, you've got 40 picograms of DNA. And you still just got one cell in metaphase 1, you still just got one cell in anaphase 1. Yes, they're being pulled, but they're still one cell. So how much present would be in each cell? Since you've only got one cell, you haven't divided yet, you would still just have 40 picograms of DNA in anaphase 1. Now, if you went all the way over to anaphase 2, where you've got the two separate cells, then it would be 20 picograms and not 40. Calculating the number of DNA molecules in a human cell during prophase 1. How many molecules of DNA are present in a chromosome of a single human cell? 2n equals 46. So in a somatic cell, it's 46. During prophase 1 of meiosis, how many would you have? Okay, if it's normally 46, you've replicated the DNA, and so when you enter prophase 2, you're going to have double because you've got the identical copies of sister chromatids and you haven't pulled the homologous chromosomes apart yet. So you're going to have double the amount you would have in a normal cell. So that would be 92. All right, a cell biologist determines that diploid cells from a particular species of Drosophila contain 16 chromatids during metaphase of mitosis. And so here's our metaphase of mitosis, and a chromatid is each of the sister chromatids. So each of the little parts of the X is a chromatid. Based on his findings, how many molecules of DNA should be present in the chromosomes of a single cell from that species during anaphase 2? So 16, if you were in metaphase 1, it would still be 16 because you've still got everybody. Because you're in metaphase of mitosis, and so you've got the duplicated. And so metaphase of mitosis is the same thing as metaphase 1 in meiosis. And so you would have 16 in both of these, but what it wants to know is how many are in anaphase 2. And so then in anaphase 2, that's where you actually get fewer chromatids. So if you go back, where you might be getting confused is this is called, meiosis 1 is called reduction division, but you're not reducing the total number of chromatids necessarily. What you're reducing is that you've split the homologous chromosomes. And so then in meiosis 2 is where you would go to the haploid state of 8. Or in meiosis 2, you would be at 8 still in anaphase 2, and then when you finally finish splitting them is where you go to the haploid state in telophase 2. So again, I may have just been confusing, but 16 means that you've got your duplicated chromosomes, because it's telling you you've got 16 during metaphase of mitosis. And so 16 means you have double, so basically 4 in here. And the same thing in metaphase 1. And what it's wanting to know is how many you have in anaphase 2. So here you split from 16 to 8 in the 2 cells, so each cell has 8. But because you haven't formed the 4 cells yet in anaphase 2, you still have 8 here. You are pulling those 8 apart, and you're only going to have 4 in each of the haploid cells that you generate. But in anaphase 2, you still technically have 8 within that 1 cell. So I hope this helps. If you still don't understand it, just let me know. I'm going to play this real quick for you.
Prior to meiosis or mitosis, DNA replication occurs. In both meiosis and mitosis, the nuclear membrane breaks down as the DNA organizes into chromosomes. In meiosis, chromosome pairs come together, or synapse, and crossing over occurs, resulting in mixing of the genetic information between the chromosome pairs. The paired chromosomes then align along the central plate of the cell and subsequently separate, one traveling to each end of the cell. In meiosis, a second division sequence occurs, resulting in 4 cells with half the number of chromosomes. Mitosis involves a single division sequence, resulting in 2 cells with no net change in the number of chromosomes.